Welcome to the multivariate EEG course. In this video, we're going to briefly review the biological basis of the electroencephalography signal and the equipment that we use to measure it. Even though most of the students of this course will already have previous knowledge of these contents, it is important to have a clear idea of what we're measuring and how we're doing it, as these factors are going to constrain the interpretations that we can make of the signal and also the conclusions that we can derive from our experiments. Electroencephalography, or EEG, is a neuroimaging technique widely used in research that has provided key insights into human brain function and its relationship with cognition. This extensive success has a few key ingredients. First, it is a non-invasive tool. EEG provides indexes of brain activity without producing damage, and this means that we can use it to study human cognition in different populations. Also, the EEG is relatively cheap when compared with other available neuroimaging methods, and thanks to this, many labs around the world can use it in their research. Importantly, the EEG offers a continuous and rich measure of neural processing, and crucially, this measure is a direct reflection of population-level neural activity. Finally, the reliance of EEG on electrical activity makes it a neuroimaging tool with high temporal precision or resolution, which is very well suited to the fast speed with which our brain analyzes information. In the next few minutes, we're going to see first an overview of the biological basis of the EEG signal, going through the types of action potentials, the summation and conduction of electricity, and equivalent dipoles. Afterwards, we're going to talk about different approaches to measure EEG and the reasons for potentially choosing between them. Action potentials are at the heart of brain functioning and at the basis of the electrical currents that we eventually measure with the EEG through electrodes that we place on the scalp. To communicate with each other, neurons release neurotransmitters of different nature, and these triggers electrical changes that, after surpassing a threshold, generate brief action potentials. These are fast or unknown changes in the membrane potentials of cells, which go from an initial depolarization to a repolarization and into a posterior refractory period in just a few milliseconds. This electricity linked to neural activity scaffolds the basis of the EEG signal. However, there are different types of action potentials, and EEG reflects only a subset of these. Neurons have three main parts: a soma, or a cell body, dendrites, and the axon, which ends in axon terminals. Dendrites are the endpoint of action potential coming from other neurons and represent the input region to the neuron. If the input action potentials are enough to trigger a response in the neuron, this generates an action potential in its axon, which travels along it until the axon terminal, where the neuron makes contact with other neurons. This is the output side of cell functioning. The presynaptic action potential is the one a neuron generates in its axon. When the inputs to a neuron induce its membrane to pass a threshold, the cell generates an action potential. This action potential is not a gradual response, but an all or none phenomenon, that is, it either happens or it doesn't. It is a very fast response, as it happens in just a few milliseconds, and the depolarization of the action potential spreads down the axon until it reaches the axon terminal, where neurotransmitters are released. We can measure these using invasive electrodes. These single-cell presynaptic action potentials are usually termed spikes and represent the output signal of a neuron. Postsynaptic action potentials are different from the presynaptic type we just saw. They do not take place in the axon, but in the dendrites of the neurons. The dendritic action potentials that reach a cell from other neurons lead to a gradual change in its membrane potential. This is a graded cumulative phenomenon, in contrast to the all-or-none nature of presynaptic potentials we just saw. Also, postsynaptic potentials last much longer than presynaptic spikes, in the order of tenths of milliseconds. They can be measured with invasive electrophysiological recordings using extracellular electrodes. These recordings show local field potentials, which are extensively used to study brain function of non-human animals. So, in contrast to presynaptic potentials or spikes, which represent the output of a neuron, postsynaptic or dendritic potentials reflect its input. As was mentioned before, the EEG signal does not reflect all electrical activity taking place in the brain, but only a part of it. With EEG, we cannot see the action potential generated by single neurons. The voltage that this generates is too small and variable to be detectable with electrodes placed on the surface of the scalp. For EEG electrodes to record a neural signal, this needs to be a summation of the electrical activity of a large group of neurons. Summation of electricity in the brain takes place in time and also in space. That is, for action potentials to be summated, they have to happen close enough both in time and in space. So, which action potentials summate? On the one hand, presynaptic action potentials do not summate. This is because they are too brief and displaced in time to allow significant temporal or spatial summation. This means that with EEG, we are not detecting the outputs that the neurons are sending. That is, we are not detecting neuron spikes. Luckily, the story is different for postsynaptic potentials. Neurons integrate graded postsynaptic potentials over space and time because they last longer, and also the arrangement allows for both temporal and spatial summation. This allows the signal of the sun dendritic potentials to be large enough to reach the surface of the scalp and be reflected on EEG data. This means that the signal that we record with EEG is similar to the local field potentials registered with invasive methodology in the brain of non-human animals, which reflects the input to the system. Even though these two signals are not exactly the same, they have similar characteristics. Not all postsynaptic potentials happening in the brain are reflected on the EEG. These have to fulfill another set of requirements. First, the group of neurons generating the source has to be large enough to reach the surface of the scalp. The neurons have to be activated in a coherent or synchronous manner. Luckily for us, neurons do not work in isolation but in assemblies, and they form ensembles through synchronization of their responses. But in addition, neurons have to be arranged in open fields for the polarities of the responses to summate. Brain neurons are arranged in open and also in closed fields. In the cortical surface, pyramidal cell neurons are aligned in parallel with each other, forming an open field. In contrast, neurons in the nuclei, such as the thalamus, don't have this parallel orientation and form a closed field. As the electrical activity of neurons has a positive and a negative poles, when neurons are arranged in a closed field, these polarities cancel each other when they sum, generating a zero net signal that cannot be measured. In contrast, the parallel arrangement of neurons in the cortex allows for positive and negative sides to summate, generating a dipole that now can be measured. Keep in mind, however, that the larger the pool of neurons involved in synchronous activity, the stronger the electrical field they produce, and thus the farther away that they can be measured. That means that a large dipole deep in the brain may still be measurable at the electrode side, although it may contribute to a smaller signal than the surface does. The neural generators of the electroencephalography signal can be modeled as electrical dipoles. A dipole is defined as a pair of positive and negative electrical charges separated by a small distance. The positive and negative side of these dipoles are the source and the sink of the current. It is important, however, to keep in mind that the polarity of the signal that we measure in EEG has not straightforward correspondence with the excitation or inhibition of cognitive function, as it depends on the orientation of the neurons generating the dipole in relationship to the scalp surface electrode measuring the signal. If the negative side of the dipole is closer to the scalp, we will measure negative voltage, and if it's the positive side near the scalp, then we will measure this positivity. The electricity travels around the tissues through conductance. The electricity of the dipole generators spreads out through the different brain tissues, meninges, skull, and scalp, until it finally reaches the E electrodes that we place on the participant's scalp. The gel applied to the electrodes helps in this process. So, taking all the previous information into account, we can say that the EEG signal reflects a summation of the active dipoles that reach the surface, which have different locations and orientations, and that summate and may cancel each other out, depending on their polarity and spatial arrangement. Because of this conduction, when we interpret the EEG signal, it is important to keep in mind that an effect generated in one part of the brain can surface in a very distant region of the scalp. So the scalp localization of our effects is a very poor indicator of the localization of the neural source. To ascertain this, we need to apply source localization techniques or use a different neuroimaging method, such as fMRI. So keep in mind that the EEG is blind to many different sources of brain activity, which you should take into account when interpreting null results of your experiments. The fact that an effect does not show up in the EEG does not necessarily mean that it does not exist. Okay, so now that we've had an overview of the biological basis of the signal we are measuring, we'll move to a few considerations about EEG measurement. To measure EEG, we need at least 2 electrodes, a reference and an active one, to record their differential voltage. In EEG research, we use more than 2 electrodes, although this number varies considerably. A classic arrangement is a 10-20 system, where 19 electrodes record EEG in specific pre-established position. The electrodes are labeled with letters corresponding to their position on the scalp: O for occipital, T for temporal, P for parietal, C for central, F for frontal, and FP for frontoportal scalp location. The number that follows indicates if the electrode location is on the left, corresponding to odd numbers, or on the right of the scalp, corresponding to even numbers. Nowadays, many labs use systems with a larger number of electrodes, that range from 32 to 256 electrodes, which are called high-density EEG systems. Although there are many more electrodes, the naming conventions remain. When deciding how many electrodes to employ in your experiment, the notion that the more the better may not always apply. You should keep in mind that increasing the number of electrodes brings good but also not that good things. On the one hand, more electrodes mean increased head coverage, which improves substantially the accuracy of algorithms of source localization. However, even for this case, improvement obtained when using 256 channels compared to 128 is only modest, which is an important issue as you are doubling the number of sensors. On the other hand, increasing the number of electrodes usually brings a certain degradation of signal quality. Having more electrodes means longer preparation times. This is something relevant to take into account, because even though modern systems have a reduced preparation time, it still takes time and effort to get a clean EEG signal in each electrode. Also, a very large number of electrodes increase the likelihood of having gel leakage among nearby sensors, and this distorts the data. A high number of electrodes most likely increase also their impedance, or the resistance that they place for conducting electricity. This leads to more noise in general and increases the amplitude of skin artifacts, such as those generated by sweat and slight displacement of the electrodes. So to summarize, when planning your experiment, it is always a good idea to consider the up and down sides of increasing the number of sensors to use. To finish, let's have a few summary ideas as take-home messages. First, EEG reflects the synchronized activity of a large pool of neurons that work in a synchronized manner and have the correct orientation to be detected on the scalp. Also, the EEG only reflects a fraction of brain activity, so keep this in mind when interpreting null results. It is also important to remember that the polarity of the EEG that we obtain on the scalp cannot be attributed to activation versus inhibition of information processing. And also, having more electrodes is not always better. If you want to learn more, you could take a look at the books by Lack and also the one by Rag and Coles for a great introduction to the technique, and also to the paper by Jackson and Bolger, which covers the neurophysiological basis of the EEG signal and measurement.